But today we started with this question right here. How many different types of gametes can be formed by individuals with the following genotypes? And I don't want to go over all of these, but I do want to take a look at this one here, and let's say this one here. Now, let me move this to the side. So if you get a question like this, so here is a genotype of an individual, heterozygous for 2 pairs of alleles. So we just want to be able to determine how many genotypically distinct gametes. So genotypically, I'm assuming there's 2 L's there, distinct gametes this individual can make. So we can do this by hand, right? Each gamete has to get 1 of the 2 alleles because of segregation, right? So we have A here and A here. This A right here, gametes that get this big A, some of them will get B, and then we'll also get a population of gametes that get A getting the little b. And for the little a, some will have to get the big B, some will have to get the little b. So we can do it sort of in our heads and just work out how many types of genotypically distinct gametes this individual can make. Now, we can also use sort of a branch diagram, a forked line diagram. So, forked line. Now, with that, we can just put the A's there. And this represents all the gametes that get a big A. And of those, some will get big B, some will get little b. Of the gametes get little a, some will get big B, some will get little b. And then we can just combine these. A goes with B, big A goes with little b, little a goes with big B, little a goes with little b. And I like this method because you're able to keep track of everything here and not make any mistakes, or you're less likely to make a mistake. And it works way better than this method, sort of in your head method, works way better when you have more than 3 pairs or 2 pairs of alleles. So the answer for this one right here is 4. But let's take a look at this individual here, heterozygous for 3 pairs of alleles. So this individual is, let's see, has this genotype. Now I'm going to use the forked line method. Of all the gametes that get a big A, some will get a big B and a little b. Same for the gametes that get the little a. And now we have a 3rd pair of alleles. So of all the gametes that get a big A and get a big B, some will get a big C, some will get a little c. And that is true for all the gametes. Some will get a big C, some will get a little c. And then we multiply across big A, big B, big C, big A, big B, little c, big A, little b, big C, big A, little b, little c, and I don't think I made any mistakes here. And we can see we have 1, 2, 3, 4, 5, 6, 7, 8 genotypically distinct gametes from this individual here. So that one equals 8. Okay, now I also wanted to look at this one right here. So very similar to the individual in C, but this one here was heterozygous for the C gene here. And this one's homozygous recessive. Okay, so the setup is a little bit different. So here's the genotype. Now, you don't put a B there. Little a here. Big B, little b, big B, little b. But this individual only has little c alleles. You can't donate a big C and a little c, only little c's. So we don't need a forked line here. We just need to put little c's. Because of all the gametes that get a big A and get a big B, all of them will have to get a little c also. There's no option for a big C. So this diagram is a little easier than the last one. Okay, so 4 genotypically distinct gametes from this individual here. Now, what if you had something like this? Well, that's a lot. That's a lot of different pairs of alleles. And it can be complicated, so we can use a formula of 2 to the n, where n is the number of heterozygous pairs of alleles. So n is the number of heterozygous pairs of alleles. And let's use some examples that we already covered. So we looked at individuals who are heterozygous for 3 pairs of alleles. And we looked at one that was heterozygous for 2 pairs of alleles and homozygous for 1. So for this individual, n equals 3. For this one, n equals 2. 2 pairs of alleles where the alleles are heterozygous. This one, 1, 2, 3. 3 pairs of alleles where the alleles are heterozygous. So to the n for this is going to be 2 times 2 times 2. So this individual can make 8 genotypically distinct gametes, and we already determined that with the forked line method. This one right here can only make 4, and we also determined that to be true with the forked line method. So this formula is very useful and helps you answer things like this very quickly, right? So we have 1, 2, 3, 4, 5 pairs of alleles here, heterozygous pairs of alleles. So the answer to this one of how many different types of gametes can be formed by individuals with the following genotypes, and different types here means genotypically different. I'm not talking about sperm versus eggs or anything like that, just genotypically different types of gametes. So this is just 2 times 2 times 2 times 2 times 2. So what is that? 2 times 2 is 4, times 2 is 8, times 2 is 16, times 2 is 32. And that's it. And it seems kind of low, but let me think this over again. 1, 2, 3, 4, 5. 2 times 2 times 2 is 8, times 2 is 16, times 2 is 32. Yep, okay, so 32 genotypically distinct types of gametes for this individual right here. Okay, so I hope you all understand how to figure that out with the formula or using the forked line diagram.